Ever wonder how a single thought, a fleeting memory, or the decision to move your hand happens in an instant? It all comes down to an incredible high-speed conversation happening constantly inside your brain. Our journey begins deep within the nervous system, a vast and intricate network of billions of specialized cells called neurons. Let's zoom in on just one of these messengers, right down to the very tip of its long arm, the axon terminal. Here, at the microscopic edge of one neuron, is where the magic of communication is about to unfold. Welcome to the synaptic cleft. This is the incredibly tiny gap, about 20 nanometers wide, separating one neuron from the next. Think of it as a microscopic moat between two castles. On one side, we have the presynaptic neuron, the sender of the message. On the other, the postsynaptic neuron, the receiver. It's across this minuscule space that all the information in your brain is passed along, from one cell to the next, in a process called synaptic transmission. Now, let's watch it in action. An electrical signal, called an action potential, zips down the axon of our sending neuron. It's like a spark traveling down a wire. When this electrical jolt reaches the axon terminal, it triggers a fascinating chain of events. Inside the terminal are tiny sacs, or vesicles, filled with chemical messengers known as neurotransmitters. For this example, let's imagine the little blue spheres are acetylcholine, crucial for muscle contraction, and the bright yellow ones are dopamine, famous for its role in pleasure and motivation. The arrival of the electrical signal is the cue for these vesicles. They move towards the edge of the neuron, fuse with its outer membrane, and release their neurotransmitter cargo into the synaptic cleft. 2. It's like a fleet of tiny boats releasing their passengers into the moat. Once released, these neurotransmitter molecules don't just float around aimlessly. They drift across the synaptic cleft on a very specific mission. On the surface of the receiving neuron, the postsynaptic neuron, are specialized proteins called receptors. Each receptor is shaped to fit a particular neurotransmitter, like a key fits into a lock. Our blue acetylcholine molecules will seek out and bind only to acetylcholine receptors, and the yellow dopamine molecules will find their own specific dopamine receptors. This binding is a moment of perfect connection, the point where the chemical message is finally delivered. 2. The precision of this system ensures that messages are sent clearly and without confusion, directing specific actions and thoughts throughout the nervous system. The moment a neurotransmitter binds to its receptor, the message is received, and the next step begins. This binding action changes the shape of the receptor, opening up a channel or triggering a reaction inside the receiving neuron. Bits creates a new electrical disturbance in the postsynaptic cell. If this disturbance is strong enough, it ignites a brand new action potential, a new electrical signal that will then travel down the length of this second neuron. And so, the message is passed on, propagating from one neuron to the next, continuing the chain of communication. This entire sequence, from the electrical signal in the first neuron to the generation of a new signal in the second, happens in less than 1,000th of a second. It's this rapid-fire relay that allows your brain to process information at incredible speeds. But what happens after the message is sent? If neurotransmitters were left in the synaptic cleft, they would continue to stimulate the receiving neuron, causing a constant, uncontrolled signal. The conversation would never end. So, the brain has some very efficient cleanup crews to terminate the signal and reset the synapse for the next message. There are two main ways this happens. The first is called reuptake. Specialized transporter proteins on the surface of the sending neuron act like little vacuum cleaners. They actively grab the neurotransmitter molecules from the cleft and pull them back inside the presynaptic terminal, where they can be repackaged into vesicles and used again. It's the ultimate recycling program. The second method is enzymatic breakdown. In this process, specific enzymes float within the synaptic cleft itself. Think of them as molecular Pac-Man. They find the neurotransmitters and break them down into smaller, inactive components, effectively neutralizing them. For example, an enzyme called acetylcholinesterase is responsible for breaking down Ds. Two processes, reuptake and enzymatic degradation, work swiftly to clear the synapse, ensuring that each signal is a distinct, short-lived event. As we zoom back out, we can see this incredible process happening millions of times a second, across the entire network. Connection. Each connection is a tiny computational unit, firing and resetting, contributing to the symphony of brain activity. This continuous lightning-fast communication between neurons is the fundamental basis of everything we think, feel, and do. It's the silent, invisible dance of chemicals and electricity that creates the reality of our conscious experience. Thank you for joining this deep dive into the brain. If you found this fascinating, don't forget to like, subscribe, and follow for more explorations into the wonders of our own biology.